Hello dear students and the members of the fraternity of Physiology and Medicine. Here is another topic in neurophysiology. It deals with the membrane potentials observed in the excitable cells, and therefore I have written the topic as EPP, EPSP, receptor potential and action potential. Well, I have written versus action potential. It's not as if this versus that. It's just the compare and contrast type of feature that I'll be discussing. End plate potential, EPP, or and the excitatory postsynaptic potential, receptor potential. These are all localized potentials that develop in the membrane of an excitable cell. And then when the cell excites, the nerve cell particularly, it develops the action potential. And when I say action potential, the membrane excitation in reality is the depolarization of the membrane. And this depolarization then travels through the nerve. It's a form of signal. The signaling through the nerve occurs in the form of action potential when we call it impulse. It's actually the depolarization which is traveling through the nerve, and this signal, the traveling depolarization, will excite another nerve, or it could be a muscle. So, when this depolarization traveling through the nerve reaches its junctional membrane, it's then going to excite the next nerve or the muscle. Particularly, I will be interested in talking about the skeletal muscle. The skeletal muscle membrane or the next nerve membrane will show these localized potentials, EPP and EPSP. Receptor potential is slightly different, so we will talk about it in some time. And therefore, let's start our discussion by saying that these are the various potentials that we see at different places during the course of excitation. That's the first generalized statement that I want to make, that these membrane potentials are observed when the membrane is going to be excited. Now, depolarization and development of action potential is described, and I like to give this example, it's my favorite example. You need to think about a firecracker. This analogy I particularly prefer because then it can explain beautifully, it can explain what happens with the membrane potential that leads to generation of excitation. So, imagine this, that there is a firecracker, an explosive firecracker or a red firecracker. Its tip will slowly burn first, and then when actually the cracker is reached, it burns in an explosive fashion. Can you imagine this? Can you visualize this? Now apply this example to our discussion over here. The burst of the firecracker is this, the action potential that develops completely at one point, and or let's say a complete depolarization. So if I were to show this graphically, the action potential is shown something like this. The nerve was resting at minus 70 millivolts, that's the resting membrane potential, and when you have applied the stimulus, the nerve is going to excite, but before that, it slowly reaches a certain potential, let's say minus 55 millivolts. This is what we are describing that happens at one point of the nerve. Let's say here is the first point on the nerve, stimulus was applied, the sodium channels, the voltage gated sodium channels, and particularly NAV 1.6 have been discovered, sodium voltage gated, that's NAV, and these channels have been found in various nerve membranes. So the sodium channels will open, sodium will enter slowly, and then when the membrane reaches minus 55 at that point, there is an explosive manner in which the membrane excitation happens and the depolarization is completed. So that point now from minus 55, it will straight away reach plus 35 millivolts, and that is what we say membrane has been completely depolarized. The membrane has excited at that particular point, and then the potassium exit will happen and the membrane will repolarize. So this is how well, potassium exit has already begun, but at plus 35 it will gather momentum. Sodium entry will stop and the potassium exit through the same point will carry the positive charges to the exterior and the membrane will repolarize at that point. However, now this depolarization which happened at one point is going to travel the next point and next point and next point will depolarize up to threshold by the positive charges that came in from the previous point. So the next point will reach the threshold due to those positive charges. The sodium brought in those positive charges, and then it will depolarize and sodium will come in from the second point and it will reach the third point, and that is how the depolarization will travel. Well, that's not the point for today's discussion. The point is that this is a full-fledged action potential. This is a full-fledged excitation of the nerve membrane, but it does not happen suddenly. You can see here that the membrane has reached threshold. That happened gradually or slowly, and then once it reached threshold, then it was completely excited. So that fact should be kept in mind. Just like the example of that firecracker, it slowly, that initial part burns slowly, and then when it comes to that end of the firecracker, then it quickly or instantaneously it bursts. That's the full excitation. So somewhat similar. Now this, in this context, let me say these are the potentials which develop locally, and they are the forerunners which eventually give rise to the development of action potential. That's the first point that I want to make, that locally at one point of the muscle, skeletal muscle, it will develop end plate potential. I'll describe it later again. And then that end plate potential will take the membrane up to threshold. The threshold will be crossed, and then a full-fledged action potential will develop in the muscle membrane and it will go into the muscle. EPSP, excitatory postsynaptic potential, is slightly different. Receptor potential, again sensory nerves, they have the receptors which develop this receptor potential, and when the receptor potential is large enough, then the action potential generated into the sensory neuron. So the first point is these potentials are the forerunners of the action potential. I mean, they happen slowly, locally, and when the membrane crosses threshold, then the action potential gets generated. So that's the first point, the membrane potentials, localized potentials versus the propagating potential, that is action potential. Now let us look at them one by one. Look, as I mentioned, the action potential travels through the nerve. What is it meant for? It is meant for a signaling process, that the nerve is going to send the signal to another nerve, or signal is being sent to a muscle for the sake of muscle contraction. That's the purpose of action potential. Now when it reaches the end of the nerve, it finds another cell over there. Now this is a representative diagram. Action potential traveling through the nerve and coming to the end. it makes a synaptic contact. The junctional region between the two cells will be called as synapse. The first cell is the nerve. The next cell also could be a nerve. So, junction between a nerve and another nerve, or junction between a nerve and a muscle. So, that also could be technically referred to as a synapse. And to make it simple, for the nerve and muscle junction, we can call it synapse, because synapse is defined as the junctional region between a nerve and another cell. That another cell could be another nerve or it could be a muscle. But to be little simpler, nerve and muscle junction, we would use the term neuromuscular junction or myoneural junction. Okay. So, even here, for the sake of discussion, nerve-nerve junction, we will call it as synapse, and nerve and muscle junction, and I am specifically talking about the skeletal muscle, we will call it neuromuscular junction or myoneural junction. Right. Now, talking about the terminology further, the neuromuscular junction, you can see that the nerve is coming in contact with a small point on the muscle. It's not the entire muscle which is being contacted by the nerve. So, that point of the muscle membrane where the nerve is making a contact, this region is called as motor end plate. Motor end plate is the region of the muscle membrane. This is the skeletal muscle. This is the area where nerve is making the contact with the muscle. And by contact, I mean the nerve is reaching there. Microscopically looking at it, there is a gap between the two membranes, of course. Right. So, this part will be called as motor end plate, the part of the muscle membrane. And the potential that develops over here will be called as end plate potential, EPP or end plate potential, the potential that develops on the muscle membrane. And which part? The part which is called as motor end plate. Why does it generate? Well, here is a motor neuron coming from the spinal cord. Look, we are talking about the skeletal muscle. So, naturally, this is the nerve that is a motor neuron coming from the spinal cord and innervating the skeletal muscle. So, this is a motor neuron. Whenever the impulse reaches its nerve ending, it is supposed to excite the muscle. I mean, that is the purpose of it. So, a chemical which is called neurotransmitter, and in this case precisely it is the acetylcholine, is going to be released. So, what happens is voltage change, the action potential, the depolarization is the voltage change that has happened. It has traveled through the nerve. It will be sensed by the voltage gated calcium channels. Voltage gated calcium channels located in this presynaptic terminal or motor nerve terminal. So, why do we need these channels? Why do we need calcium? Look, first of all, listen to this part carefully. This is a vesicle, the acetylcholine vesicle, and this is the nerve membrane, and you know already that all the membranes of the cells are negatively charged, the cell membranes and the membranes of the organelles. So, even the vesicular membrane will be negatively charged and the nerve membrane also will be negatively charged. But we want that whenever the impulse comes here, the acetylcholine should be released, and for that this vesicle with acetylcholine should fuse with the nerve membrane. If both are negatively charged, they will repel. I mean, there will be a repulsion. And therefore, we need that there is a bridge formed between the vesicular membrane and the nerve membrane, the presynaptic membrane. For that bridge formation, we need the calcium. So the job of calcium is to make that bridge happen between, and of course there are synaptic proteins involved in this bridge formation. That's not the topic for today's discussion, but we need calcium for this fusion and eventual exocytosis. But calcium cannot be kept inside the cells. You know that the calcium is kept very, very low intracellularly. Cytoplasmic calcium has to be very, very low. So therefore, this is the arrangement. Now coming to the real point. Our real point is not this. The real point is about EPP and EPSP. So let me bring your attention to the real point. That is, when the impulse arrives, it will be sensed by these voltage gated calcium channels. Calcium channels will open. Calcium will come in to the terminal, and then acetylcholine vesicles will migrate towards the membrane. They will fuse with the membrane, and acetylcholine will be released into this so-called synaptic cleft or gutter. Now this acetylcholine will combine with its receptors on the motor end plate. In the case of neuromuscular junction, the receptor is the nicotinic receptor, nicotinic acetylcholine receptor. And now you should start concentrating particularly on this part. This nicotinic acetylcholine receptor is a non-specific cation channel. Non-specific cation channel. That is, it can allow sodium as well as potassium to move through the channel. There is a receptor portion. Let's just draw it here. There is a receptor portion with which the acetylcholine will bind, and there is a channel portion, and it's a ligand gated ion channel. It's a ligand gated ion channel. The ligand acetylcholine will combine with the receptor part, will bind to the receptor part, and then the channel gates will be flung open. And as it opens, now listen to this part. As it opens, it will allow sodium and potassium to move through the channel. So, sodium can come in through the same channel, and potassium can go out. You know, they will move by their concentration gradients. So, sodium will come in, potassium can go out. But the point is, we want the membrane to depolarize. Let me just bring your attention to one more point. It's not the main feature, but just you can make a note of this. The events that are happening at the synapse or the neuromuscular junction, they are electrical, chemical, electrical. The electrical action potential will release the chemical, which is the acetylcholine, and that chemical will again generate an electrical response in the muscle membrane or even in the nerve membrane in the case of the synapse. So, it's a kind of transduction mechanism, it's a transducer mechanism, that one form of energy is getting converted into another form of energy, right? So, but that's besides the point. Just make a note of this, that electrical action potential releasing the chemical, and that chemical is going to generate the action potential in the muscle, and the muscle will contract as a result. Now, we want the muscle to depolarize, because I mentioned that excitation means what? Excitation means depolarization. Okay, the membrane should depolarize. If sodium comes in and potassium going out, it happens at the same time with same amount, same quantity. then the membrane will not excite. But that's not what is happening. Listen to this part. The muscle is resting at minus 90 millivolts before the impulse came. The muscle is resting at minus 90 millivolts. Now, when the channel is opened, acetylcholine is released and channel is opened. Sodium comes in and potassium goes out through the same channel, but sodium entry will predominate over potassium exit. I mean, if I were to give you hypothetical figures, 100 sodium ions will come in and only 5 potassium charges ions will go out. Why is that? That is because, listen to this part extremely carefully. I have discussed it in a previous video called as equilibrium potential. So listen to this again. Muscle is resting at minus 90 millivolts. And at this voltage, if you allow both sodium and potassium movement, sodium movement will be very, very large as compared to the potassium movement. Why is that? Because sodium has got an equilibrium potential of plus 61 millivolts. Potassium has got the equilibrium potential of minus 96 millivolts. Membrane is at minus 90, which is close to minus 96, the equilibrium potential for potassium, but is far away from plus 61 millivolts, which is the equilibrium potential for sodium. So the point is, the ion movement will depend on this particular factor. Note this point again. If the membrane potential, like in this case minus 90 millivolts, if the membrane potential is close to the equilibrium potential of a particular ion, then that ion movement will not be great. The ion movement will be small. And if the membrane has moved far away, I mean the membrane potential is far away from equilibrium potential of an ion, then that ion will move in large amounts. Okay. Because after all, what is equilibrium potential? That potential on the membrane at which there is no net movement of that particular ion. That's the equilibrium potential for an ion. Right. But this principle you must never forget. So, coming to the point, minus 90 millivolts, now the channel is opened, sodium enters, potassium goes out, but sodium entry predominates over potassium exit. And therefore, there is a net entry of positive charges, net influx of positive charges, which will result in depolarization. That was one point I wanted to make. Another point is, when one impulse, for every one impulse that comes to the motor nerve terminal, for every one impulse, there is almost release of 60 vesicles of acetylcholine. Every impulse will cause almost 60 vesicles of acetylcholine. And each vesicle contains about 10,000 molecules of acetylcholine. The point is, there is a massive release of acid or a large amount of acetylcholine will be released with every one impulse. Each impulse which is going to cause the contraction of the muscle is going to cause large release of acetylcholine. So that another point should be noted. And when such a large quantity amount of acetylcholine is released with one impulse, with every impulse, it causes the sodium influx, which is a predominating event, and a local potential develops. And this local potential is called as end plate potential. As I mentioned, the potential that develops on the motor end plate. This is naturally a depolarizing potential. We are looking for excitation of the membrane. So, excitation means depolarization. Inside of the membrane should become positive. That's depolarization. So, this is a depolarizing potential. And this potential has got a value of about 40 millivolts. That means 40 millivolts of depolarization will happen locally at one point of the muscle membrane. It will happen locally. And it means what? From minus 90 millivolts, where the muscle was resting, 40 millivolts depolarization means it will go to minus 50 millivolts, minus 90 to minus 50, the 40 millivolts depolarization, positivity. It means the muscle membrane will definitely cross the threshold. You remember what I said in the beginning. For the action potential to be generated, a certain threshold potential has to be reached. It will reach slowly, the threshold will be reached, and then it will burst into the action potential, that particular point. So, now bring that point here. A 40 millivolts depolarizing potential has been generated locally. And it was generated like that, as I mentioned, the firecracker example, slowly burning the first portion, and then threshold was reached and a quick burst of the firecracker. So, something similar. Slowly it developed, and one point of the membrane from minus 90, it reached minus 50, 40 millivolts of positivity has come in. I am just speaking in a simple manner. So, this point will now cross the threshold and it will convert into an action potential. And once action potential is generated, please remember, it's a self-propagating and self-regenerative type of event. It will then propagate by itself into the muscle, and the muscle, as a result of this, the muscle will contract. So, this is end plate potential. But the point that I want to highlight is that what is the magnitude of end plate potential? It's a synaptic potential. It is developing on the motor end plate. It's a forerunner of the action potential, and its magnitude is 40 millivolts. The point is that one impulse, when it reaches the motor nerve terminal, look, why it has reached here? Because the muscle needs to contract, the skeletal muscle needs to contract. So, when even a single impulse is reaching the motor nerve terminal, the meaning is cortex has taken the decision, motor cortex has decided that this particular muscle should contract at least once. I mean, when one impulse is sent, at least there should be one contraction by that. And therefore, such a large release of acetylcholine and leading to a large influx of sodium, therefore resulting in a large end plate potential, a local potential. It means muscle will definitely reach and cross the threshold by that impulse, by the impulse which has come in. Muscle will definitely generate one action potential, and muscle will definitely contract once. You understand the point? The point is entirely about the magnitude of end plate potential. If you compare this with EPSP, excitatory postsynaptic potential, here is a synapse between a nerve and another nerve. Let's say a synapse in the CNS, nerve junction. Impulse arrives at the nerve terminal, the presynaptic terminal. Calcium channels open, okay, that's also common. And then neurotransmitter vesicles. Now here it's not only acetylcholine, there are hundreds of neurotransmitters discovered. But we are talking about the excitatory neurotransmitter, the glutamate, the aspartate, the common ones. Well, the neurotransmitter vesicle will fuse with the presynaptic membrane and exocytosis will happen. Neurotransmitter will be released. It will bind with the receptor on the postsynaptic membrane, and the channel which is associated with this receptor. Now this time it's not a non-specific cation channel. We are just comparing now. Look, EPP, it's a synaptic potential seen in the muscle membrane. EPSP, it's a synaptic potential, postsynaptic potential seen in a CNS synapse. Excitatory postsynaptic potential, that's EPSP. So, now this channel in the case of a postsynaptic membrane in the CNS, these are specific channels. Unlike there in the muscle, that was a non-specific cation channel, these channels are specific cation channels. So, either it could be sodium channel or it could be calcium channel. In the case of glutamate, it's known to open the calcium channels. So, but this channel is specific and it will allow that specific cation to enter through this channel. And when this cation enters, there is first development of a localized potential, okay, which we now call as EPSP, excitatory postsynaptic potential. What is the amplitude, magnitude of EPSP? It is less as compared to the EPP. The magnitude of EPSP was very, magnitude of EPP, I beg your pardon, was very large, 40 millivolts. What was the purpose of that large magnitude? This is a repetition, I know, but just a quick. What was the purpose of that large magnitude? That the muscle membrane should definitely reach threshold and should definitely convert into one action potential. Why was that the purpose? The purpose was that because motor cortex had taken the decision that this particular muscle should contract once at least. So, one impulse sent, and or it could be more than one impulse. Definitely every impulse is going to lead to contraction. That's the point I am making. Impulse sent from the motor cortex must result in the muscle contraction. For that to happen, muscle membrane should excite, and for that to happen, the EPP magnitude should be large enough. Only then the muscle membrane can cross the threshold. But compare this with EPSP. Again, this is also an excitatory potential, okay, excitatory postsynaptic potential. That too, EPP was also an excitatory potential. It was causing depolarization. What's the difference? Difference is in the amplitude. It's just about 2 to 4 millivolts of depolarization. Entry of sodium or calcium ion will locally cause a local depolarization, a local small depolarization of 2 or 4 or 5 millivolts. So, this nerve, postsynaptic nerve, which was so far resting at minus 70 millivolts, that's the RMP for the nerve, can at the most depolarize from minus 70 to minus 68, let's say, or minus 65. 5 millivolt positivity or 5 millivolt positive potential developed means this point of the nerve from minus 70 can go up to minus 68 or minus 65. But the point is that enough to generate action potential? No, it's not enough. Obviously, let's say, see, we have said already that the threshold should be reached. Let's say that for a nerve, its threshold is, this is a graphic depiction, minus 70, minus 55. Let's say threshold is minus 55. Only when the nerve reaches minus 55, the threshold is reached and enough number of cations will come in after that and depolarization will be completed. That's not happening here. It happened in the case of muscle, but it's not happening in the CNS synapse. So the point is, single impulse in the presynaptic neuron will not cause excitation of the postsynaptic membrane. Excitation means generation of action potential. It can show depolarization of 2 to 4 millivolts, but it's not reaching the threshold by itself, and therefore it will not convert into an action potential. So then, how is it that impulse from the presynaptic nerve goes into the postsynaptic nerve further? How does that happen? It will happen by summation. Summation of EPSPs will be required. Single impulse cannot excite the next nerve. But now let's have a diagrammatic representation. Let's say there are 3 presynaptic terminals ending on one postsynaptic membrane. Then, and the impulse coming in each of this presynaptic terminal, causing a 5 millivolt depolarization locally. So, one impulse coming in this neuron number 1, it causes 5 millivolt depolarization by opening of sodium channels and sodium entry. Similarly, another neuron sending the impulse and causing 5 millivolt depolarization over here locally, and the third impulse coming in the third neuron, 5 millivolts. Now these 5 millivolts are EPSPs, excitatory postsynaptic potentials. They are excitatory because depolarization is happening. And then there will be summation, addition, to be precise, to be simple, addition of 5 millivolts. By the way, this will be called as spatial summation. So all the potentials that develop on this entire space, they will get added when they are happening within 15 milliseconds of each other. We will discuss about summation later, but that's 5, 15 millivolts. That means now this neuronal membrane, postsynaptic membrane, which was resting at minus 70 millivolts, will now reach minus 55. Threshold is reached. Well, I have taken the hypothetical figures. Let me tell you that these are the hypothetical or theoretical figures just for the sake of explanation. So, minus 70 is the figure, RMP of the nerve is minus 70, and minus 55 is the threshold, that also is the figure. But apart from that, these figures are just for the sake of explanation, 5 millivolt for instance. So, from minus 70, the nerve, this postsynaptic nerve now will reach minus 55, threshold is reached, and that will lead to one impulse, one action potential in the postsynaptic nerve, postsynaptic neuron, and that impulse will go down in that next neuron, postsynaptic neuron. So, did you notice here that EPP, one impulse in the motor neuron, will cause one impulse in the muscle and result in one contraction, because motor cortex had taken the decision that this muscle has to contract. Whenever I send the impulse, the muscle has to contract. Therefore, EPP was large. It was definitely one EPP was converting into one action potential. But in the case of CNS synapses, that's not happening. There are multiple neurons firing at the same time, and all those EPSPs are getting added to then convert into one action potential in the postsynaptic neuron. The reason that I feel here is that our CNS is a dynamically evolving structure throughout life, and it is continuously working on the basis of a let's say trial and error type of method, and therefore that certainty has not been provided. There are lots of checks and balances in the CNS synapses so that it does not fire unidirectionally, achieving one single target at a time. So it will always work like this. Small potentials called as EPSPs are small potentials. They will generate due to the impulses, multiple impulses, and that that will eventually convert into one impulse in the postsynaptic neuron. So that's the difference between EPP and EPSP. Why do I have to compare this? Is because both are postsynaptic potentials. Okay, but the difference is this. I mean, both are going to excite the postsynaptic nerve. EPP will excite the skeletal muscle, EPSP will excite the postsynaptic neuron. But that is a critical difference between the two. That there the impulse is a definitive signal to produce a definitive result, muscle contraction in the case of EPP. Here in the case of CNS synapses, it would be a constantly adjusting nature of the CNS synapses, constantly learning nature by trial and error. Therefore, one impulse will generate only a small excitatory potential, and therefore multiple excitatory potentials have to add up to form a signal, definitive signal in the postsynaptic neuron. So that's why I chose this topic particularly to explain the comparison between EPP and EPSP. Well, so far as the receptor potential is concerned, receptor potential is seen in the case of the sensory receptors, and the receptor potential amplitude, let's say here is a Pacinian corpuscle, a typical receptor, and a sensory neuron that emerges from within the corpuscle and will send the signals to the spinal cord. So, a mechanical stimulus is applied. Now, basically, receptor potential is also a depolarizing potential that develops locally. So, let's say here is a part of the skin where a mechanoreceptor is present, and these are the channels. Now, these are again, because I am talking about the mechanoreceptor, this is a mechanosensitive sodium channel or mechanically gated sodium channel. Remember, in the case of muscle or in the case of CNS synapse, those were ligand gated ion channels. The non-specific cation channel in the case of muscle, the specific cation channel in the case of CNS synapse, it could be either sodium or calcium in the case of EPSP generation, and here the mechanically gated sodium channel. So, you apply mechanical stimulus, the channel opens, and there is influx of the cation, influx of sodium, right? This is a part of the skin. So, influx of sodium and a certain localized potential will develop here. Locally, the potential depolarizing potential will develop. So, this part is called as generator potential, rather this generator region. Just this region where receptor potential is going to be generated, and then a correction. Okay, this part is where receptor potential will be generated. So, it's called as generator potential and generator region, and the first node of Ranvier is going to develop the action potential or spike. So, that is called as spike generator region. Spike generator region. Well, then which is this region? Yeah, it's called as transducer region. Transducer region. As I said previously, this region, well, you know, in the case of synapse also and neuromuscular junction, we have seen already that there is a transduction, electrical impulse releasing the chemical energy, and that again converting into electrical impulse in the next nerve. Even here, there is a transduction. We applied a mechanical stimulus, particularly in this case. Mechanically gated sodium channels opened, positive charges have flown in or positive charges have come in through the channel, and a local depolarizing potential has got developed, local electrical potential has developed. So mechanical energy converted into electrical energy. What is that? Transduction. So this is called as transducer region. Now when this electrical potential develops, we will call it receptor potential. A small localized potential developed on the receptor membrane. And when it's more than 10 millivolts, then it will generate the action potential, it will generate the spike at the first node of Ranvier. So transducer region for the receptor in the skin, we applied a mechanical stimulus, and that will be converted into electrical potential, let's say a local depolarizing potential of a few millivolts. When it is more than 10 millivolts, then an action potential will be fired because of the influx of cations, because of the influx of sodium ions. Action potential will be finally fired in the first node of Ranvier, and then it will travel through the neuron to the spinal cord and to the cortex where we feel that touch. The point that I am making is, what is the amplitude of receptor potential? The key point that I wanted to make. What is the amplitude of receptor potential? It depends on the intensity of the stimulus. If the stimulus is too faint and very few sodium ions and positive charges enter, then it may not generate the action potential in this sensory neuron. So receptor potential, its amplitude has to be more than 10 millivolts. And therefore, anywhere between 10 millivolts and 100 millivolts. Receptor potential amplitude is anywhere between 10 and 100 millivolts. It depends on the intensity of the stimulus. Greater the intensity, greater will be the amplitude of the receptor potential, and of course greater are the number of impulses that are generated through the sensory neuron to make the cortex aware of the sensation. So that's the receptor potential. Again, localized potential, but it gives rise to the action potential which will then propagate. Okay. Well, the only exception, now this is an important point, the only exception to this is, and I am just writing, though this is not a part of today's discussion, but I am writing it here, the rod in the retina, the receptor for vision, the rods and cones. The rod receptor potential, that is when light reaches the retina, rod will develop the receptor potential. It's the visual receptor. Rod receptor potential, please remember, is a hyperpolarizing potential and not a depolarizing potential. So a great exception to this rule, hyperpolarizing potential. That's the exception, because so far we have said wherever there is a stimulus, whether it was EPP or EPSP or receptor potential, all of them are depolarizing potentials. Membrane will depolarize slowly first, and then when membrane reaches threshold, action potential will be generated from those potentials. The exception is this: when light, the stimulus for the rod, when light reaches the rod in the retina, rod will not depolarize. Its receptor potential is not a depolarizing potential, it is a hyperpolarizing potential. Why is that? And what is the logic behind it? What are the evolutionary reasons possibly behind it? We will discuss. That will be the matter for some other video. I'll take it up later on, that the logic rests on evolution as well as the physiology and the channels and so on. So we'll discuss it in another video, of course. my current discussion, our current topic revolves around the localized potentials, EPP, EPSP, receptor potential versus action potential. So that's the receptor potential. Finally, let me compare these membrane potentials. So receptor potential or EPSP or even EPP for that matter versus action potential. How do you compare them? And mind you, this table, if you by heart for your prof exams, that will be good enough to fetch you marks. So just by heart this table. If you have a short note on EPSP or if you have a short note on receptor potential or EPP, you can just write this table. It will be good enough. So first thing, EPSP or receptor potential or EPP, these are localized depolarizations, localized potentials. They are confined to that region. Okay, there is a passive flow of charges from that region for some distance, but that's it. What I'm trying to say is, it's not a propagating potential, it's not a traveling potential, whereas action potential is a self-propagated potential. Means when the action potential forms at one point, then you don't have to do anything, it will travel all by itself. Again, take the example of the firecrackers. Let me tell you this. If imagine that there is a line of red firecrackers. This is my favorite example, so I'm just coming back to it again. Imagine a line of red firecrackers. You will just give fire to the first cracker. It reaches threshold, and after that you don't have to do anything because first cracker reaches threshold and bursts, and after that the firecrackers will go on bursting all by itself, because first cracker while bursting, it passes the flame to the next cracker. So next cracker automatically reaches threshold and bursts. So one point developing action potential is good enough, then the action potential travels by itself. So in that sense, it's a self-propagated process. It propagates, whereas these are just localized potentials. Second point is, these are decremental potentials. Decremental potentials. What I mean is, let's say an impulse came. This is a CNS synapse, nerve junction. One impulse came, sodium channels were opened, and a small 5 millivolt depolarization was developed locally. Now, if nothing else happens, means another impulse does not come, then this will exponentially decrease and it will decay and completely disappear by 15 milliseconds. So the summation which I told you some time back can happen between two impulses and two EPSPs if they are occurring within 15 milliseconds of each other, because this potential is a decremental potential. And when I say decremental, it means in terms of time, it means it in terms of the distance. I mean, the charges will come in and the charges will passively flow over a certain distance. So in that sense, whatever was the strength at this point, at one point where the channels have opened, the strength will decrease from that point towards the distance on either side. So in that sense, it is decremental. The strength will decrease on either side, plus over time, over 15 milliseconds, it will exponentially decay and disappear, completely stop. So next impulse, if it has to generate one more EPSP, it has to come within 15 milliseconds of the first impulse. Only then the summation can happen, and that's called, by the way, that's called as temporal summation. Temporal means time. Summation, I'm going to describe synapse properties in the next video. So, right, so decremental, whereas action potential is a self-regenerative process and it is a non-decremental type of potential. Non-decremental type of potential, meaning, let's say here is a nerve. we have applied stimulus at the first point of the nerve, and this point generates action potential, fully excited, depolarization from minus 70 to plus 35 millivolts. This is the full amplitude. Okay, so now it is going to travel. Okay, it's a propagated potential. Fine, it's going to propagate, it's going to travel. At every point, it is going to develop with same amplitude. No matter what is the distance it is traveling, no matter what is the length of the nerve, at all the points it will have the same amplitude. With distance, it is not going to decrease in amplitude, and therefore I said it is a non-decremental type of conduction, non-decremental type of potential. It's a self-regenerative process. At every point it will generate itself. So it's an automatic way, in a sense, once the threshold is reached at one point. So non-decremental conduction, at every point it will be minus 70 to plus 35. Even last point also, when it excites for this nerve, it will be from minus 70 to plus 35. No decrease in strength. Just like the line of red firecrackers, even there the first cracker bursts with the same intensity of sound, and last cracker also will burst with the same intensity of the sound. It will not be a decrease in intensity of sound. Of course, it should be of good quality, but that apart, so similarly, even the action potential is non-decremental type of conduction. Third point, and I think most important point. So listen to this part. That's the most important comparative feature, and that is these localized potentials. The localized potentials, they are called as graded potentials. That's a very key difference. Graded potentials means what? I am taking hypothetical values. Suppose one impulse comes in the presynaptic neuron and it releases 2 vesicles of acetylcholine or 2 vesicles of neurotransmitter, an excitatory neurotransmitter. One impulse, 2 neurotransmitter vesicles released and developing a local potential of, let's say, 2 millivolts. I am just simply taking hypothetical values at this stage. If 2 impulses are coming in the presynaptic neuron, so 4 vesicles of neurotransmitter will be released and 4 millivolts depolarization will happen on the postsynaptic membrane. The EPSP will be of 4 millivolts. If 3 vesicles are released, then 6 millivolts EPSP. 4 vesicles are released, 8 millivolts EPSP generated. I am saying in the sense that it shows gradations. It will depend on the number of vesicles released and therefore the number of positives charges that enter the postsynaptic membrane. It shows gradations. Compared to that, the action potential, which obeys all or nothing principle, all or none law. Yes, it obeys all or none law. So far we have seen 3 features, 3 comparative features between the localized potentials and action versus action potential. Action potential obeys all or none law. I mean 0 or full. That means no gradations. Action potential does not show gradations. Of course, that is when all conditions are same, action potential will be of the same amplitude. If the conditions change, the AP amplitude can change. But the point is, whenever it develops for that particular condition, its amplitude will be full. So 0, if the membrane does not reach threshold, no action potential will be developed. 0 response, the membrane does not excite. And if the threshold is reached, then full action potential develops, its full amplitude develops. So 0 or full, nothing in between. Again, the example of firecracker will suit to understand this. If that initial portion, initial thread does not completely burn and reach the cracker will produce 0 sound. But if that threshold is reached, I mean that thread is burnt completely and then the cracker bursts, it will burst with full intensity. So zero sound or full sound, nothing in between. Similarly, action potential. Zero response if your stimulus is good enough, is not good enough. I mean, if the stimulus is too weak, then zero response. If the stimulus is strong enough to reach the threshold, to membrane to reach the threshold, then action potential develops fully. There's nothing in between, no gradations. Whereas EPSP, receptor potentials, they will show gradations. I mean, receptor potential, if your stimulus has a weak intensity, only few sodium channels will open, less number of positive charges will go in, and small receptor potential will develop. 2 millivolts, 4 millivolts, 6 millivolts, 8 millivolts, that's possible. So that is gradations, graded. And the last one, and then we will close this particular topic. Last differentiating feature is that these localized potentials are said to be monophasic potentials. Monophasic potentials. That is a single phase of depolarization of 2 or 4 or 6 millivolts. Single phase. That's it. So if I were to say EPSP, it's 4 millivolts of depolarization. That is what we call as EPSP. Monophasic, single phase. Whereas action potential is said to be a biphasic response. Now, I am not going into the details of biophysics when I say biphasic response. To put in simpler terms, I would say electrically two opposite types of events constitute the action potential. The depolarization and repolarization. Electrically two opposite phases. I mean, if this is a nerve and it's resting at minus 70 millivolts, inside of the nerve becoming positive is called as depolarization, and again the inside of that point goes back to negative, that's called as repolarization. So electrically two opposite types of events, depolarization, inside becoming positive, and repolarization, inside again goes back to negativity. So that's a biphasic response. I've put this part simple without using any terms of biophysics or physics, but these are the comparative features between the EPSP or receptor potential or EPP versus action potential. So in case if you have a short note on EPSP or receptor potential or action potential, you may refer to this table and that will be good enough. So that's the topic which I wanted to discuss today. Well, just a quick revision. These are the localized potentials that develop when stimulus has arrived a particular point, and when the membrane reaches threshold, then they convert into the full-fledged depolarization and action potential. So these are the forerunners of the action potential. First they develop like the thread burning up to the firecracker, and then the cracker bursts. The action potential develops fully in a very quick explosive fashion. That was the topic for today's discussion.